Thank you, Jason. I'm happy to be here today with you and tell you a story about drug development and biomarkers and how they interact. And part of the moral here will be often a very concrete method applied to a very simple example can give results that, whether or not they lead to a clinically used biomarker, nevertheless can change the climate and get your colleagues to think differently about using multivariate biomarkers. So the story starts with this compound, apparently not with a laser pointer though. The story starts with a compound here called this rather non-euphonious name. It has the property that it inhibits a protein called MDM2. And MDM2, as most of you probably know, is itself an inhibitor of p53, which is a tumor suppressor gene. So if you are a tumor trying to exist in a context of wild-type p53, you need to shield yourself somehow from the action of p53. And one way to do that would be to upregulate or overexpress or amplify or somehow otherwise increase the function of MDM2. That will then shield you from the effect of p53. So this compound is actually very specific and it's highly targeted. Obviously, the mechanism is intimately intertwined with cancer, and so we're looking at looking for a patient population that's going to have a wild-type p53 context and amplified MDM2 tumors. Now, if you do that, you find some interesting things. Number one is you get lots of slices of other indications. You get 10% of glioblastoma, a few percent of breast, a few percent of liver cancers, a few percent of kidney cancers, a few things like that. It turns out that this particular genetic context, wild-type p53, amplified MDM2, is a molecular characterization that does not map well onto the sort of histological, anatomical, historical diagnostic categories. There's no one disease that says, this is our guy, with one exception. And here's the exception, taken from Oncomine, which is actually taken, of course, from the Baratina data and from the Vasilek data. This is dedifferentiated liposarcoma. And what you notice there immediately is this is basically each bar here is a patient, and the vertical axis is essentially this is the log of the number of copies of the genes. And basically about 90% of the patients have 4 or more copies, and about 2/3 of the patients have 8 or more copies. So this looks like this would be a great context for our compound. And I showed this to a pathologist and said, gee, it looks like 90% of the patients have MDM2 amplified, and his response was, yeah, and the other 10% are misdiagnosed. Pathologists always have the good jokes about that kind of stuff. But basically, this is the set of patients in whom our drug has to work. And indeed, when you try that out, if you look at some cell lines that are derived from liposarcomas, you have pretty high potency. This is about maybe 100 nanomolar or something. You're getting by the time you get to 1 micromolar, you're getting pretty much a complete cell kill, and you're getting apoptosis induced at similar places. Now you might think at this situation, okay, we're done, right? We have a compound that's highly targeted. We have a mechanism that's intimately intertwined with the cancer. We have a patient population. That's pretty much all you need, right? The problem comes when you say, well, how many patients are there anyway? And I was just looking up the numbers a few hours ago. It turns out that there are, order of thousands, 5,000, 8,000 something liposarcomas in the US every year, and the rate across the world is actually pretty similar. Of those, about 1/5 are dedifferentiated, so you're looking at like 1,000 patients a year. Now remember yesterday somebody showed that really frightening graph from Scannell's Nature Reviews drug discovery paper that showed the number of drugs per $1 billion has been going down on a log linear plot for the last 60 years. It's very expensive. And so this is very frightening to say if that's the only patient population, this is just not economically feasible. We'd have to do something else here. So what we want to do is we want to say, can we break out? It's fine, we're going to test this. And indeed there is a phase 1 trial which is going on in this disease right now using this compound as a single agent. And that's all fine. I can't tell you the results yet, but you can imagine they're not going to be too bad. But we want to try and break out of the context in which we require MDM2 amplification to predict a patient population. So we want to search for a marker in the space where we want to look for wild-type p53 patients, but who are also wild-type MDM2. They're not amplified. Are there anybody there? Are there any patients there who will also respond? And we knew that there were lots of cell lines that would do that. So we said, okay, fine, let's go look across a wide panel of cell lines. We found a panel of 66 wild-type cancer cell lines across a pretty wide variety of cancers. And what you're seeing here in the graph is each dot is 1 compound cell line pair. There are 4 compounds, as shown in the legend down here. The green and the yellow ones are 2 competitor or academic compounds, and the blue and the purple ones are our compound and its backup. And what you're seeing on the graph is, on the horizontal axis, the GI50 for cell count, the concentration at which you're going to get half the growth inhibition. And on the vertical axis is the concentration at which apoptosis is 5-fold induced. So they're both potency-like axes. The contours are probability density contours computed by kernel density estimation, basically convolved with a Gaussian. And that's meant to convince you that where I've drawn these 3 red circles, that's a reasonable way to draw cuts in there and say there's sort of 3 different response classes. Now you can do this, in a more statistical way, but it's just a graphical way to look at it. What that tells you is there is one cluster up here at 10 micromolar in both directions that says basically it doesn't respond to anything. You put the drug on those cells, they don't care, they ignore you, they go on having their party, and that's really bad. But these other 2 groups show us that there are 2 different response classes. These cells down here respond with apoptosis. Okay, we're killing the cells, they're going down, we have a GI50, everything's great, and they're showing an apoptosis marker, so we actually know how they're dying. So we know they're dying, we know how they're dying, that's great. There's another group up here at the top, the top cluster, which are dying, but they're dying without apoptosis. And we don't know what that's causing. I'll tell you what's causing that in a little bit later. But basically it says we have 3 response classes, and the thing we'd like to know is there a marker which can tell us the difference between the non-responders and the responders? If you look only at cell count, here in this graph I'm showing you the GI50 for cell count on the horizontal axis and the maximum effect on the vertical axis, the efficacy, the Emax. And what that tells you is a couple of things. Of the responders, first the thing that we liked was our compounds were about 10 times more potent. That was great. The med chemists are really smart. They can do a lot of stuff for you. But on the other hand, what they cannot do is increase the efficacy. Once you've inhibited the target 100%, you get all the effect and that's it. You just don't get any more effect, right? So it says that, yes, indeed, we do seem to have multiple response classes. You either respond or you don't. You either have apoptosis or you don't. And we're not getting anything over and above the general effect of the target, but we are improving the potency a little bit. So let's see if we can find something that distinguishes the responders from the non-responders. So the simplest kind of enrichment analysis, just a Mann-Whitney statistic, told us that among the responders, hematological cancers were in there, melanomas were in there, Differentially liposarcoma was in there. That had better be in there, otherwise there's something wrong with the whole enterprise. And one of the things I try and encourage sort of more junior colleagues to do is introduce tests of your work at every possible opportunity to make sure you haven't lost the thread. Because oftentimes you'll transform your data until finally all the information is gone, and now you're fitting on noise. So we keep testing things and say, yeah, okay, we should be able to rediscover dedifferentiated liposarcoma, and we did. We also discovered that the resistors were enriched for uterine and cervical cancers. We don't really have a theory for that, or we have a theory for that. We don't even have any evidence. It might be oncoviral. Those are oncoviral induced, and there's a different mechanism for inactivating p53. So basically we have some evidence here based on a fairly massive dose response experiment that there are some cell lines which we know are responders and some which are resistors. So what can we do with that? Well, we can do this. We did a bunch of gene expression. Now this is actually fairly old work, about 3-4 years old now, so it's Affy chips, and you'll see there's some other more traditional methods here. We took the exact cell pellets that were used in the dose response experiment and put them into a gene expression experiment, because one of the things we found was that the cell lines acquire mutations. Just because Cosmic tells you that something is p53 wild type doesn't mean that the cells you have in your hand are in fact p53 wild type. The cells are alive. They go and they do things when you're not looking. And in fact, we found later some of the cells had mutated. Okay, so we have a bunch of Affy chips, and first we drop all the Affy controls, then we drop the genes that aren't called present at least a couple of times, then we drop the ones that aren't very variable, because if they're just on constant, that's fine. All of this can happen outside of a cross-validation loop. Then inside a cross-validation loop, we start z-transforming, doing a t-test, and saying which genes are different between the responders and the non-responders. That gets you a certain number of probe sets. Then for each probe set, you pick the best one for each gene, and that leaves you with 39 genes. All right, so these are genes that can be tested. And this is all done under a cross-validation loop. Interestingly, you might notice that there is no separate arm here explaining why there was apoptosis in some cells and why there was not in others. The reason is you get no features here. And the reason it turns out is because the assay was actually not very good. They measured apoptosis at one time point at 72 hours, so the cells that apoptose early don't leave any signs behind. So that was more of an assay artifact. So in what remains, we'll just look for, is there a difference between the responders and the non-responders? So the bottom arc is under a cross-validation loop. It gave us 39 genes, and we want to know, can we use those to classify things and predict who's going to respond, who's not going to respond? Okay, so when you look at that, what do you get? Well, here on the rows are the genes, and on the columns are the samples. And again, we're trying to see if unsupervised methods, just by clustering, can recover the responder versus non-responder context without knowing the training labels. And in fact, they do. These guys over here at the bottom, the color bar at the bottom here tells you these are the non-responders and these are the responders over here. And unsupervised learning can indeed recover the distinction that we were trying to make, even though it doesn't know what distinction we're trying to make. Now, it didn't do it on the first split. The first split in the column dendrogram actually is something else, but it's an unsupervised method. It doesn't know what we care about. But we did recover things, and we see a couple of interesting things here. Number one, in the set of predictors are the drug target, MDM2. That's nice. It would be nice if the target was a predictor. If the target is up or down regulated, that ought to affect whether or not the compound works. CDKN2A is also in there. And what's nice about CDKN2A is it's a downstream transcription target of p53. So if that's upregulated, then p53 is already in good shape. Hence, if we bring p53 back in, it's not going to do any good. But what's really interesting to look at is, if you look at the responders over here, they're nice and clean. You know, red, green, not much stuff. Or these are the resistors. But if you look at the responders, they're all over the place, all kinds of noise. Usually you would think that there's one way to respond to the drug, which is to go by the mechanism of action. There's all kinds of ways to evade it. But this is the reverse. And what's going on here is we have a signature for resistance to the compound. And after you dig in a little bit, you find this is essentially a signature for p53 inactivation, which actually makes a great deal of sense because that's our compound's mechanism of action. Okay, so when you do gene set enrichment on this set of genes, you find that it's enriched for the things that we expect, CDKN2A pathway, MDM2 binding, p53 binding. We did this enrichment not because it tells us anything new, I mean it might have, but because again we're checking to make sure we didn't lose the thread. You know, does the data still make sense after you've done all these selections? So you know, we're always checking our work and validating things. So we have this 39 gene set. What can we do with it? Well, we'd like to regress on it and try and build a classifier, but you know, 39 parameter classifier, that's kind of a pain. I don't know what happens in Toronto, but on the streets of Cambridge, if you say you've got a 39 parameter model, people are very suspicious of you, properly so. So what we did was singular value decomposition and looked at the principal components. What you're seeing in this heat map is on each row is a principal component, going from the first to the last, and on each column are the cell lines. The black ones are the resistors, the white and gray ones are the two classes of responders, and what you can see is the color green is associated with the resistors, and the other colors are associated only with responders. You can make that quantitative and statistical and do a t-test and everything, but basically what it tells you is only the first principal component matters. And in fact, when you plot them in the space of the first three principal components, the resistors stand out. So then we did a logistic regression on that first principal component. That gave us a statistically significant model. If I were going to do this again, of course, I would use Lasso and GlimNet and things like that. But again, this was a couple of years ago, so I just did principal components. When you use the loading matrices to map back to genes, you get a 39 parameter model, even though you've only fit one gene. And the result is you get 39 genes. As you might expect, the ones that are resistant have the the ones that are associated with response have a negative regression coefficient. The ones that are associated with resistance have a positive regression coefficient, and they're the ones you expect. MDM2 is in the set, but it's not the strongest predictor. And this is sort of a tug of war we often have with biologists. The gene that is most intimately involved with the disease process is often not the gene that produces the strongest signal. Our strongest signal is something else. Okay. Last point, when you look at the principal component scores, the red bars here are for the resistors, the blue bars are for the responders. You can see that they're very clearly different. This is what we use to convince biologists that, you know, maybe we should do something other than just measure the phosphorylation state of your favorite gene. A multivariate classifier can actually get you a much better answer. The thing that we did, this is the probability of being inactive as a function of the principal component score. And what you notice is it's a very strong step function. It's very decisive. It's never sort of in between wishy-washy. And it's 94% accurate under threefold cross-validation. That's what we use to convince clinicians that this is worth paying attention to. Okay, what have we learned? We've got this compound. It inhibits MDM2. It's highly selective. We found that it worked very well in dedifferentiated liposarcoma, but we want to break out of that context. When we do that, we find that there are wild-type p53 and wild-type MDM2 tumor models which fall into three classes, actually two classes once you get rid of the bad assay about apoptosis, and that we can actually predict those with a 39 gene marker. Now, what happened to this? Well, This probably won't be commercially developed because after this we went and we don't need a marker for the differentially liposarcoma application. If you have the diagnosis, that is the marker. On the other hand, we went and did some synergy experiments with this based on some hope from the marker and discovered that there was another compound, a MEK inhibitor, which exhibited very strong synergy with us in a RAS-activated context. And so there's actually another trial starting exactly this month, so it's now public knowledge, which uses wild-type p53, wild-type MDM2 RAS activation as its marker. So the main impact of this work wasn't that I suddenly got people to use a 39-gene marker or that we could have used LASSO to compress it down and made a PCR plate and commercialize the diagnostic, but rather that it motivated other research to find other contexts in which there was a simpler marker that we could generate with next-gen sequencing, and that's what's being used in the clinic. Thanks very much. Oh, by the way, this is poster SB03, so it's back there. If you have questions, grab me and I'll take you through the details.
We have time for one quick question, otherwise we'll, in the interest of time, go to the next speaker. If there's a question, please go to the mic.
Thank you. I'm curious, how did wild type or more primary non-transformed cell responded to this compound? And did they look more as the responders or non-responders?
It depends which gene you're talking about. If they're p53 mutants, they are almost always resistant. All right, p53 is the sine qua non of any response.
But the wild type non-cancerous ones?
The non-cancerous ones respond far less, and the reason is they're not dependent on have so much MDM2 around that they can completely shut down p53. So when you shut down MDM2 in those cell lines, they're perturbed, but they're not terribly perturbed. And indeed, that's borne out by the sort of the tox results in the phase 1 trial as a single agent.